Good morning, everybody, and thank you very much for joining our SPHN webinar. Today, we will hear about a sort of a special project because it is quite complementary to the other SPHN driver projects we have. As for example, it does not run over our central biomodality infrastructure or contribute to the SPHN dataset. The Create PRIMA project, meaning clinical research from multi-modality big data sources without proprietary interfaces in a multi-center approach, will analyze data from 4 cantonal hospitals, namely the Cantonal Hospital in Aarau, the one in St. Gallen, Ente Ospedaliero Cantonale in Ticino, and the one in Basel-Land. The CREATE system is an innovative approach to the management of complex unstructured data and aims to enable interoperability between systems and organizations, basically, so to say, in parallel to the SPHN interoperability strategy. So we are very curious to hear about the progress of this project. And with this, I would like to welcome our speaker of today, Professor Dr. Jörg Leuppi. Jörg is a professor of internal medicine at the University of Basel, heading the University Clinic of Medicine at the Cantonal Hospital Basel-Land in Liestal. So I'm happy to pass the word to you, Jörg, now. Thank you very much for taking the time and being with us today.
Thank you very much. Good morning, Catherine. Good morning, everybody. Thank you very much for this very nice introduction. Actually, gave a perfect summary of our project. But now I would like to go through our ideas. I would like to summarize some words about our mission. I would like to mention our consortium, like you already mentioned, the 4 hospitals working together with an IT company. I'd like to give some background, mention some features, milestones, and then I really want to go into the research projects which are currently started to prepare, timeline, project status, overview, and of course also challenges we are facing day by day. CREATE, as you mentioned, Catherine, the CREATE system is an innovative approach to the management of complex structured as well as unstructured data. The project will enable interoperability between systems and organizations and exchange of nationwide routine clinical data, which will be accessible for research. Now our consortium, as you mentioned, is based on 4 cantonal hospitals. There's one in Tessin, Bellinzona. There's one in St. Gallen. There's one in Aarau, and is our hospital, Kantonsspital Basel-Land. We really would like to work together, and we are aiming at the end to analyze data of 4 big hospitals. The current main applicants can see on the main left side. You see that's people from our hospital, like our cardiologist, Gregor Leibengut, but also I really would like to mention Giorgia Lutti-Corridori. She's doing her PhD within that project, and she's one of the main important people here running that study. On the other hand, we also have co-applicants, the representative for the other cantonal hospitals like Professor Paul Haslo and Dr. Angelika Hammerer from Kantonsspital Aarau. Then we have Professor Michael Brendle from Kantonsspital St. Gallen, and we have Professor Luca Gabutti from Kantonsspital Bellinzona in Tessin. And our project, this driver project, is in very close collaboration with the industry. We have a very important project partner, that's a company called Iterata from Canton Aargau. And these people are helping us to perform that data search and at the end also help us to do the analysis of the data. Some thoughts about the background. You all know it's very difficult to access large clinical data sets. And you also know it's not easy to access these data. If you work in a hospital, perhaps you have your local way to save the patient's data. You write the history, you write the information about what's going on. Then you have a program which all the laboratory data are saved. You might have a program who have the radiologies, pictures saved and the reports saved. But often it's nearly impossible to write the questions like you want to know all the patients with heart failure and you want to have from all these patients simultaneously laboratory values and maybe information about the radiology. That's not possible. All the data are here. And the data often varies in different formats. And predominantly, the data are saved in an unstructured form like reports. And the challenge with regards to linking these different data sources for profiling individual patients or patients group is a big challenge. So with the principle and features of our Create Driving project is to manage complex, structured and unstructured data. We do not want to copy or move data. We just take a reference of the data. We want to create searches and algorithms which are applied to these data. We want to reduce all the interfaces. And of course, we want to have a high level of data security. And we want to force interoperability between systems and organizations, which finally should be suitable to search and analyze big data. Where are we now at the moment, or what's our milestones? 2017-18 mainly, we wrote the proposal, and we're very thankful and glad we got the grant approval end of 2018. 2019 was a quite intensive year to prepare in collaboration with lawyers within the 4 hospitals and of course with the company to set up contracts. So we have now a contract between the 4 hospitals, the consortium contract, and this consortium has also a contract with the iterator, with the software company. That's quite a delicate, interesting, but also a time-consuming process to have a very proper, clear and contract, which finally we were able to achieve. So we're very thankful for having now a nice consortium which is organized within that contract. So therefore, more or less a year ago, exactly actually, we officially started with our driver project, the Create project. Within this year, we mainly invested time to implement the system in the 2 cantonal hospitals, Basel-Land and Aarau. We also invest a lot of time to set up the dictionary and search profile for our 6 research questions. We started to collect data and we on the way to improve our searches and our data collection. And one of the important parts at the moment is data validation. So we have to make sure that we really find all the data we want to find and reduce false negative and false positive data results. Next year, we will concentrate on the implementation of that system on the other 2 hospitals in the Kantonsspital St. Gallen and in the EOC in Bellinzona. And because in Tessin people mainly speak Italian, we also have to set up a dictionary in Italian language. We have to improve the dictionary and we have to improve the search profiles for our research question also in these 2 hospitals. And then we continue to collect data within Aarau and Basel-Land, but also we want to improve the data collection within St. Gallen and Bellinzona. And we also have to improve and validate our data collection. Then the last year, 2022, we want to improve and finish the interoperability. We want to share the results. We want to even work harder at the analysis of our data. And of course, we want to write publications. Our CREATE projects bases on 6 research projects. There's on one hand the so-called DECODE project, which we would like to characterize patients admitted to hospitals with dyspnea. The second one is the influence of thyroid hormones on clinical presentations in patients with coronary heart disease. Thirdly, we interested to identify cases of Methotrexate-induced pulmonary toxicity in a fully searchable routine clinical database. Fourthly, the POC heart failure trial means a serial point of care BNP measurement for therapy monitoring during hospitalization patient with acute decompensated heart failure as a therapy monitoring during hospitalization, which is a prospective unblinded randomized controlled pilot trial. Fourthly, we want to look on general clinical practice in requesting redundant laboratory tests. We would like to eliminate the famous twins or triplets. That's a huge problem in the hospital data sets, that patients and data are stored several times. And lastly, we want to develop a risk score to predict Metamizole-induced leukopenia. Now, the timelines so far. So within the first year, we started with the DECODE. The DECODE will be a project will last over 3 years, but we started now in our hospital and in Aarau to find these dyspnea data and started to analyze them. We also started within these 2 hospitals, actually with all 5 or 6 questions, although the famous twins, we just started now. In the second year, we will continue in KSBL and Aarau, but also want to start, of course, in St. Gallen and Bellinzona, and finally want to finish it everywhere in all 4 hospitals in the third year. Now, I would like to come a bit more closer to the different studies or projects. The status at Basel-Land at the moment, so we have the IT system more or less implemented. The DECODE project, the characteristic of patient admitted to hospital with dyspnea, we looked on 1 year so far. We started to search for data from 2014. This is completed. We did analysis to perform, to calculate sensitivity and specificity. And the manuscript, mainly about the method of this study trial, is currently under review. The POC trial, the heart failure trial, using BNP value to decide which is the way to improve the treatment. In Basel-Land, we have completed 50 patients, so the pilot trial has been finished within that year. That was a huge achievement. We now currently analyze this data and prepare the manuscript. In Basel-Land also we started to collect data about the Metamizole-induced leukopenia, so the data collection has started at the moment. Allow me to talk a bit more in detail about the DECODE project. The characteristic of patient admitted to our hospitals with dyspnea interests us. Dyspnea is a common and a major cause for emergency department visits. The etiology of the reason why dyspnea occurs is huge. Probably 30 to 40% is caused by respiratory diseases, rather 30–40% caused by cardiac diseases, and then something between 10 to 30% have other reasons. And not seldom, our multi-morbid patients have lung and heart diseases which cause dyspnea. So little data actually available on its frequency and causes, especially of this multi-morbid patient within Switzerland. Now the aim of the DECODE project is to collect epidemiological hospital data about frequency and cause of dyspnea. We would like to characterize these patients' population by collecting data on different examination like lab values, imaging, ECGs, etc. We want to perform cluster analysis to identify patient characteristics at the time of hospital admission to guide early targeted diagnosis and identify patients who respond to guidelines recommended therapies. Now, one of the challenges is actually to create a dictionary. The language we use about dyspnea is quite a huge range. We use different language terms. And then also the challenge what we really write down. One question is what the patient says, and then finally at the moment what the physician writes down. And so we had to decide which dictionary we really want to use, which words we want to use, and which words we want to search within our database. So we had finally an automatized screening with the CREATE system of the available documents by using a dictionary. And then we decided to use 1 year. Just by random, we decided to use year 2014. And then with 2014, we had a manual search. So our researchers sat down and went into the computer program where we save our patient data and did a manual search to look for patients which came into our hospital, were admitted to our hospital with dyspnea. Parallel, we performed a search with our CREATE system and then started to calculate sensitivity and specificity. And at the beginning, we had a sensitivity of less than 30%. So we had to improve. We had to find better search strategies. We had to work on the dictionary, etc. So currently, that's the current status of our DECODE from 2014. You see all the dyspnea really admitted to our hospital with dyspnea, and you have all the patients which didn't have any dyspnea, and then we had a sensitivity and specificity between 75 and 84%, which is an accuracy of 83%. So it's still not perfect, but I think it's quite useful level that we can start to do further analysis. Now, within the DECODE, we also want to look on specific diagnosis. So we will in the future look on heart failure, we want to look on COPD exacerbation, we want to look on lung embolism, but we also want to look on pneumonia. And patients hospitalized with pneumonia are usually treated with intravenous antibiotics. And it's often safe to switch to all treatment within a few days of hospitalization once the patient doesn't have any fever anymore and his condition improves. An early switch to oral medication has the advantage to shorten hospitalization time and causes less health costs. So an optimized use of antibiotics can also help to limit resistance development. And so our question was the therapy of hospitalized pneumonia patient guideline conform? At which day of hospitalization was the administration of antibiotics changed from intravenous to oral, where intravenous antibiotics continued longer than clinical indicated? So actually we do like an audit. We want to look how is our performance in our hospital. Do we do that, what is recommended by international guidelines? So far, we had a search. This time we chose 2019 and looked for all patients admitted to our hospital with pneumonia. Up to now, 762 of approximately 1,050 documents were checked. We found 537 patients with pneumonia. We had 225 cases with a so-called false positive result, and we have a correct result in 70.5%. So what's the sources of error within this 225 so-called incorrect documents? Now the computer looks for the word pneumonia. And if somebody writes history of pneumonia within the diagnosis list, it will be called as a case. Then pneumonia mentioned in medical evaluation or maybe written is included. It's found in 79 cases. Then it's somewhere written pneumonia as a differential diagnosis, as another diagnosis was mentioned. Then maybe incorrect spelling. So Klebsiella pneumoniae, Klebsiella pneumonia. Then recurrent pneumonia, but not acute pneumonia. Lung fibrosis or possible pneumonias were all terms which caused incorrect case finding. I'd like, unfortunately in German, but I show you some real-time or real-life examples of wrongly diagnosed pneumonias. So history of pneumonia written in the diagnosis list, and then somebody gave a summary of the CT scan, and the radiologist wrote no segment pneumonia found, and the computer sees the word pneumonia. Or later on in the Nebendiagnosen, other diagnosis, you have status or history of pneumococcal pneumonia. Then somebody suffering from a cancer and had in the past a history of post-stenosis or cancer-induced pneumonia, and then the computer found that word as a pneumonia. Or somebody wrote, if within the text pneumonia has been excluded, and the last pneumonia was found or occurred a year ago, so both words were falsely detected as a real pneumonia. Or misspelling, so instead of Klebsiella pneumoniae was written Klebsiella pneumonia, and then the computer detected that as a case. So we have to work on the case findings, we have to work on the dictionary, and we have to program that the computer knows what he has to exclude and not accept, and what he has really to accept as a case. That's the status at the moment. Now the Metamizole, using Metamizole as a painkiller, it's a very good medication, a good drug. However, rarely, but sometimes very severely, people can develop a leukopenia during treatment with this medication. And Metamizole, as mentioned, is a good analgesic and antipyretic drug. Metamizole treatment bears the risk of blood disorders, including agranulocytosis. Only little is known about underlying risk factors for developing leukopenia under this metamizole treatment. One previous study of 57 cases and 139 controls identified co-medication, history of allergy, and hepatitis C infection as a risk factor, but it's very small numbers. Risk factors can help to identify patients for whom metamizole should be avoided, and the determination of possible risk factor in patients who have developed leukopenia in association with metamizole treatment by comparing them to patients who have not occurred that side effect is a study design we would like to do, a so-called case control study. So we start to analyze retrospectively data of cases with leukopenia associated with metamizole and of matched controls who did not develop leukopenia after metamizole treatment. Collected patient characteristics, laboratory data, data of medication use will be collected. And we want to, as mentioned, compare these cases with matched controls to identify risk factors for developing leukopenia associated with metamizole. So far, our steps currently, we try to identify the patient cohort, we search for matched controls, and we extract patient characteristics, laboratory results, and use of medication. So at the moment, we have a broad search terms in order to maximize sensitivity, and we screening of all possible available documents. So the search terms we use is the brand names and the generic names like Novalgine, Metamizole, Minalgine, Novamine sulfone, and the association with leukopenia, achronocytosis, neutropenia, and we found so far 746 cases. If you then go into these 746 cases, which reported both metamizole and leukopenia, we had to exclude the majority of the patients because patients did not use actually metamizole use, although the word was found in the document. Patient did not have a leukopenia, although it was mentioned, probably again, history of leukopenia or history of metamizole. Then patient with metamizole use after leukopenia onset, so it's history. Patient with metamizole use as required, and patient with leukopenia not due to metamizole, because perhaps they had had a chemotherapy as a reason for the leukopenia. So far, we left with 295 patients with leukopenia associated with metamizole use. Two projects which has been started in Canton Spital Aarau, and therefore the IT system has implemented as well in Aarau, and they have started the Methotrexate project. That's the question of Professor Hasler, identifying cases of Methotrexate-induced pulmonary toxicity in a fully searchable routine clinical database. So the data collection has been completed and the analysis so far performed in Aarau. So we want to identify cases of Methotrexate-induced pulmonary toxicity, and the scientific background is low-dose Methotrexate is widely used as an immune suppressor agent. Undesired effect of Methotrexate is frequent, like headache, abdominal discomfort, increased liver values, but it's also potentially life-threatening conditions such as cirrhosis, cytopenia, and pneumonitis. So Professor Hasler is interested on real side effects of pneumonitis. The reported occurrence of Methotrexate-induced pneumonitis in a case series in the literature varies between 0.3 till 11.6%. Reviews with estimated mortality rates of cases with potential Methotrexate-induced lung toxicity goes up to 13%. So he did the whole population of analysis of enterprise search engine addressable data of 15,000 cases, searched for terms indicating pulmonary toxicity according to the KSBL criteria, a junction by expert pulmonologists considering findings of high resolution CT lung scans and bronchial alveolar lavages, 88 age and sex-matched exposed control cases with no indication of pulmonary toxicity was found, and then a descriptive statistic and exact logistic regression has been performed. So that's the flow. We had he found in Aarau nearly 2,700 cases where methotrexate is mentioned. Nearly 1,900 methotrexate was really exposed to this patient, and the full criteria was in only 14 cases found. 12 cases with a defined pulmonary toxicity and 2 cases with a probable pulmonary toxicity, and then with the judgment also from the pulmonologist and rheumatologist, they finally had these 12 cases with defined pulmonary toxicity out of 1,887 methotrexate exposed people and 2 cases with a probable pulmonary toxicity. So where are we in overall? You see the full bullet means we have achieved what we need to achieve, and then if the half bullet is half full, then we have achieved half. So in Basel-Land, we have the system readiness actually achieved. We find the cases now. Now we have more work on the dictionary and improve the correct cases. The data readiness is done. The interoperability of the results is half done. The search projects are running. We still clearly haven't finished them, and we haven't done any, we've just submitted 1 publication. We're aiming for 2021, of course, to fulfill the gaps, with the end that in 2002 publications are done and everything is working KSBL. In Aarau, the main work will then be done in 2021. So next year is a big Aarau year. So far, the lab is connected. The system readiness is done for rheumatology and laboratory, but we need to have access to full clinical data, so we're working on that. St. Gallen will mainly start, system readiness will be done within 21, and in Bellinzona we will start in 21, but the main thing will be done in St. Gallen, and Bellinzona will be done in 22. So the challenges are mainly the semantic. You know, we have to the language we use, the language which is written down, and the spelling, all that is it's quite a huge challenge that we really find the right cases. We live in a 4-language country. At the moment we run the study also in Tessin, so we have the translation into Italian, sorry. So this translation from languages will be a challenge, and hopefully one day we can also have hospitals with French-speaking area. Then of course we have to do the translation French as well. A very interesting but really challenging situation are the communication between different stakeholders. You know? The IT experts, the doctors and the researchers use different languages. So we have to achieve that what physicians would like to know and how they formulate, that researchers really also understand what they have to analyze. And the biggest challenge is that IT people understand what we like to get. and that we speak the same language and that we do the same search at the end. So that's really interesting, but also challenging to achieve that. And the challenge suitable for detecting a cohort, but more difficult for retrospective research. You know, we live in a real-world situation and we really work with real-world data. That's interesting, but challenging. Our system will be very good for future studies if you want to case finding, if you want to just include maybe patients with a high BMP, so the system always finds the value and then it shows up the patient with a high value, and then we can include that patient. That will be very easy done. Now to analyze retrospectively, to find the right documents in the right place, in the right words, it's really challenging. But that's our job. We really want to achieve that within the next 2 years. So that was an overview where we are at the moment. I think it's a very interesting and great project. And I'm very thankful for all the people involved in that study. I'm very thankful for the SPHN to finance and help us to perform that driving project. I'm very thankful for the company Iterata, who helps us to really do that study. But the biggest thank goes to the research group, who really work very hard together with these IT people to have this study going on and to finish it within the next 2 years. So thank you very much for the attention. I'm very happy to answer questions. Thank you.